Partillion Bioscience, an American company has developed a new tool to expand your functional toolkits. It's called the Nanovials, and you will see there are a lot of applications. It's a very, very interesting technology. And without more waiting time, I will pass the microphone to, is it Zuli or Katie who will start?
It's me. It's Zuli.
Okay, I let you go. Thank
Thank you so much. So just to introduce myself, my name is Zuli Peralta. I'm a senior product manager here at Partillion Bioscience. Also joining me today is Dr. Katie Kershaw. She is a senior scientist at Partillion Bioscience as well. So I'm going to get started with the first half of our talk to show you how nanovial technology can be used to expand our functional toolkit. And in this presentation, we're really going to cover nanovial essentials and their applications. So we all know about the genome, which stores genetic information and acts as the blueprints for living organisms. These blueprints are transcribed into RNA, which act as messengers, carrying these instructions to the cellular machinery that translate these into proteins, which are the building blocks of life. And although these proteins build up a cell, a subset of these are secreted. And these play a crucial role in intercellular communication, tissue regeneration, and disease progression. And these secreted proteins can provide valuable information for things such as drug targets or drug candidates, which can greatly benefit human health. Now, over the past decades, flow cytometry has played a pivotal role in expanding our understanding of biology. Traditional flow cytometry really centers on the analysis of cell surface receptors. But there exists an equally vast array of secreted proteins which are significantly understudied. These things such as cytokines, antibodies, and other bioactive substances really play key roles in cellular communication and physiological process. However, up to now, researchers really don't have reliable ways to sort single cells based on their secretions. We also have new advancements in single cell technology that really help us understand the heterogeneity of a sample of both cells and proteins. So whereas before we were able to just take bulk measurements and not really be able to look at this heterogeneity, what single cell allows us to do is isolate individual cells and probe them and better understand either their transcriptome, their genome, or their proteins by being able to compartmentalize them into single cells. And this has allowed researchers to find rare cell types that sometimes drive disease or specific phenotypes. However, when it comes to profiling single cell secretions, the existing methods that are out there really lack sensitivity, often destroy the cell, or rely on expensive instrumentation. And we think it would be really useful to isolate cells based on their secretions and to be able to assess cell-to-cell interactions in a single cell level. And our answer to this are nanovials. These are sub-nanoliter containers for functional single-cell biology. What you can see here in this cartoon is that they have a semi-enclosed cavity. They hold less than a nanoliter of volume. In the video that I will show you, hopefully it runs, they are optically transparent and are made up of a hydrogel, which means they're highly modifiable. And because they have an open cavity, they can easily go ahead, we can easily exchange reagents, as well as be able to perform biochemical reactions within these nanovials. And as you can see here, they're really, really tiny, so we're able to fit millions of these nanovials in a single tube. We have two formulations of nanovials. We have our original formulation. These are biotin related, which means they can be easily modified. They have low autofluorescence. They're really great for complex fluorophore panels. And most recently, we've introduced our EZM nanovials. These are also biotinylated. Again, you can easily modify them. And this new formulation has cavity localized extracellular matrix, which is ideal for adherent cells, improved cell recovery, and for high secreting cells to reduce the level of crosstalk. Now, how do we obtain single cell readouts? We do this in a variety of ways. First is the cavity size actually restricts multiple cell loading. In our most commonly used formulation, the nanovial is about 35 micrometers in outer diameter and has an 18 micrometer cavity. So this allows, it fits most of the cells that are out there. We also have larger cavities that are available for multi-cell assays. Another way that we ensure single cell readouts is by loading nanovials in excess to cells, and this reduces the likelihood of 2 cells being in 1 nanovial. Our suggested ratio of cells to nanovial is 1 to 5, and we can also tune this ratio to further optimize based on your specific application. As I mentioned, these nanovials are really easily modifiable to capture whatever your cellular secretion of interest is. So in this example, you'll see a cartoon of a nanovial, and within the cavity, we have millions of biotin binding sites localized to the cavity. Again, this is a depiction of our ECM formulation. Also within the cavity, we have extracellular matrix, ECM, that allows binding of adherent cells. So using biotin strip tableting chemistry, we can go ahead and modify these with different capture moieties of choice. So this means we can use antibodies against cell surface markers. We can use antigen CAR to see our binding to capture cells, or we can easily just use the ECM that is within the cavity to capture other sensitive adherent cells. And as you can see here, we've demonstrated capture of various different cell types. Another neat thing about the nanovial technology is that after you run a nanovial workflow, your cells are still alive and you can recover them. So in this picture, you'll see that after we've run a nanovial experiment, we can recover the nanovials. So you'll see the nanovial here. You can see the secreting cell, and you can see this fluorescent signal from the secreted antibodies. And you can see a little bit more in this depiction of the cartoon of what's actually within the nanovial. So once we are able to recover them post-sort, we can simply put them in some growth media and allow them to expand. And as you can see here, now these cells have outgrown the nanovial itself, meaning that you can recover whatever cell that you've specifically selected for, and then use these in any downstream assays. Apart from preserving the cell viability or being able to recover cells, we've actually seen that the cavity has some sort of protective effect on some of these cells. So to show you this, in this experiment, we took cells and we took one portion of them and loaded them into nanovials. And we took another portion of them and did not expose them to nanovials. We stained both of them with CD138 APC and then sorted them and measured their viability. From this plot, that you can see is that out of all the hybridoma cell lines that we tested, the ones where the cells were loaded into the nanovials have higher viability than ones that were just free cell suspensions. And we believe that this is because nanovials act sort of like a little helmet on the cell to protect them against shear stress as they go through FACS sorting, really maintaining that cell viability. Again, nanovials are a really awesome tool to allow you to do sensitive functional assays to capture and measure secreted molecules for individual cells. As I've shown you, they're non-destructive. They allow you to maintain the cell's high viability to allow for downstream applications. And one other thing is that you don't need any new equipment. So you can analyze and sort in a high throughput manner using your existing flow cytometers. You can apply these in 2 different ways. One, you can build your own assay. So you can purchase biotin-related nanovials only and use your existing antibodies or antigens. And this is really awesome if you've already, if you're working with a very niche cell type, or if you've done a lot of development into picking out antibodies specific to your cell type. You can then just buy the nanovials and perform a nanovial workflow within your existing reagents. Again, we have them available in 2 formulations, our original formulations, which are biotin-related and work really well when you're doing a lot of complex fluorophore panels. And we have our ECM nanovial formulation, which have extra features for adhering cells and reducing some of that crosstalk if you have very high secreting cells. Another of our offering are ready-to-go nanovial kits. And these include antibodies for common cell types and their secreted proteins. Right now, we are really focused around our antibody discovery kits. These are built off of ECM nanovials and are mostly focused on mouse. Again, these are ready to go workflows. We have user guides, we have some videos, and really to get you started as soon as possible. We also have cytokine kits. These are early access, so these are on a case-by-case basis. If you are interested in trying out some of these cytokine kits, please reach out to us so we can discuss product fit. I briefly mentioned this earlier. We have 2 nanovial sizes for different applications. And for most applications, we are using our 35 micron outer diameter nanovial. However, we do have larger nanovials for large cells or for multi-cell assays, which is something that we're currently developing now. And for both of these were engineered to be compatible with most sorters, analyzers, and single-cell sequencing technologies. Do note that some of these will require some adjustments for some instruments in order for you to go ahead and sort these. And we have guidance to help you do that and do some gating as well. Now I will turn it over to our senior scientist, Katie Kershaw, to go ahead and tell us a bit more about the nanovial workflows and all the applications that you can perform with them.
Great, thank you so much, Zuli. Just a quick housekeeping check. I want to make sure the slides are advancing. Can everyone see the slide that says simple benchtop workflows?
Yes, we can.
Perfect, great. All right, good to go. So this slide is really here to show you how easy the nanovial experiments can be. If you know how to pipette and you know how to centrifuge, you're ready to do a nanovial assay right now. So the first part of our assay is the instrument-free portion. And this can be performed in a biosafety cabinet or wherever you normally do your cell culture work. So the first step after you've modified your nanovials will be to simply pipette your cells into those nanovials to load the cells. And again, the cell capture antibodies will load those cells and only load the cells that you want. In the second step, this is your secretion incubation, so you allow the cells that have been captured to secrete whatever you're interested in them secreting, and that will also be captured on the nanovial by your modification. To visualize that, you go to step 3, where you'll stain with a fluorescently labeled antibody against your secretion of interest. And this is very similar to how you would do a traditional flow staining. In fact, you can even work this secretion detection stain into your existing flow panels. And then you would move on to the instrument portion, where in step 4, you can sort and analyze based on that secretion of interest on whatever your favorite flow cytometer is. And as Zuli mentioned, those cells remain viable in the nanovial. They're even protected from the forces of sorting in the nanovial. So you can do further downstream characterization, such as these growth assays, or even put them through single cell sequencing or other sequencing workflows that you already have. And we have many external partners, both in industry and academia, who have validated several workflows with the nanovials, and there's just a sample listed here. And we'll go through some versions of these applications in more detail in the following slides. But a high level, we've done a lot of antibody discovery workflows, cell therapy workflows using T cells, and also sequencing-based workflows, both single cell, and you can also do traditional RT-PCR. So for our very first in-depth application, we'll look at antibody discovery. And this workflow was done in collaboration with Alloy Therapeutics. They are a biologics discovery contract research organization in the States. And their antibody discovery arm was very interested in using plasma cells as the source of their antibodies. Of course, plasma cells, they're often called antibody factories. They secrete hundreds to thousands of antibodies per second. But the challenge can be that they downregulate their BCR, and so it can be hard to know what their antigen specificity is. Alloy was very interested. They had this hypothesis that antibodies developed from plasma cells would be better quality and have higher affinity than those from traditional memory B cell workflows, but they hadn't had a way to really study that yet until they partnered with Partillion and developed this nanovial assay. So they modified their nanovials with a biotinylated antigen against which they were raising antibodies. And that was able to specifically capture only antibodies that were antigen specific. They then had their fluorescent detection antibody to stain those nanovials that had the secreted antibody on the surface. And as you can see in this flow plot, they had really nice separation of their antigen specific cells. Here it's a FITC anti-IgG antibody, and you can see this is all from loading plasma cells. So they have their B cell markers that they did in the same panel as their secretion staining. Again, these cells remain viable throughout this whole assay, so they sort them out. And then they did their traditional like heavy and light chain sequence recovery workflow and re-expression to do further characterization. And some of their characterization tests are shown on the right. So they're looking at affinity here, both protein binding and cell binding. And hopefully what you can appreciate is that they got some very nice binders out of this plasma cell assay. There was even a picomolar binder highlighted here, which is very exciting. But also exciting for them was that they could validate their hypothesis. As you can see in this chart, on average, the affinities of the antibodies developed from plasma cells were much higher affinity than those from their traditional memory B cell workflow. In our next application, we'll focus on T cells. This work was done in collaboration with Owen Whitty's lab at UCLA, and they're looking at TCRs here. One of the hurdles to TCR therapy is that the TCRs with the best affinity may not necessarily be the ones that are the most potent, the most cytotoxic, the most functional. And traditional assays to look at TCR mostly includes tetramer staining, and tetramer staining relies on very high affinity. So you might miss out on lower affinity TCRs that would be very potent inducers of cytotoxicity. Using the nanovials, they were able to combine the binding with the functional assay so they could look at these more interesting TCRs. And they did this by essentially turning the nanovial into an artificial antigen presentation cell. So they modified the surface of the nanovial with biotinylated peptide MHC complexes. And since there are hundreds of millions of binding sites on the nanovial, you get hundreds of millions of binding sites for peptide MHC. And this increases the avidity, which allows you to enrich for TCRs with lower affinity, which again might be the ones that are the most potent. So in their assay, they were able to enrich these TCRs using the peptide MHC, and this also activates the T cells that bind. So then you can allow them to secrete. In this case, we were looking at interferon gamma as our marker of cytotoxicity, which was captured on the surface of the nanovial with an anti-interferon gamma antibody. They then use their existing surface stains for CD3 and CD8, in addition to their fluorescent detection stain for the interferon gamma, and we're able to sort out these cells. And in these images, you can see that the CD3 and CD8 staining is localized to the surface of the cell, but over here, there's a nice one where you can see the purple interferon gamma staining all along the cavity of the nanovial. And when they compared this nanovial-based workflow to their traditional tetramer workflows, one, they were able to do much higher throughput. So they could start with a lot more cells because they're enriching using the TCRs on the nanovial. And as a consequence of that, they pulled out a lot more cells. They were able to sequence more cells. And in the end, they got almost five-fold more complete clonotypes. So more diversity with the nanovial staining compared to the tetramer staining. So this allowed them to, again, increase their throughput and also find more diverse TCRs. In a very similar workflow, we'll now look at CAR T cells. And this was developed in-house at Partillion. Here, again, very similar, you have the antigen modified on the nanovial that the CAR is against. And this not only binds the CAR T cell, but also activates it. So then you can allow it to secrete and capture that on the surface of the nanovial to not only know that the CAR T binds, that it's specific, but also that it is functional. In this assay, we also used interferon gamma as our marker of cytotoxicity of CAR T functionality, and we sorted the cells out using a Sony. And then, as you can see, we gated on the cell-loaded nanovials and then looked at their viability with a calcine stain. And then if you drill down to look at the interferon gamma secretion levels, you can sort out these high secretors and confirm, as you can see on these images, the pseudo-colored red interferon gamma staining all around the surface of the cavity. Now, this told us that the CAR Ts were binding and that they were activated. So they're functional. And traditionally, how you would look at this would be a bulk assay such as ELISA. And that's what we did here. As you can see in the blue, we have our CAR T cells, a control versus one stimulated with its antigen, which was CD19. And there was a large fold induction of interferon gamma. However, when you look at the single cell resolution that the nanovial assay allows you, you can see that these high levels of interferon gamma are really driven by a few cells that are prolific secretors. But if you just looked at the average value, it's not really representative of the whole population biology because it's a minority of the cells that are responsible for this large level of interferon gamma secretion. So again, bringing up the non-destructive nature of the assay, you can sort these cells out and you can do further downstream characterization. So say, for example, you want to study these high secretors versus these low secretors and figure out what is driving these changes in secretion. And that is actually what our next application was interested in. So they were looking back at antibody secreting cells or plasma cells. And this was done by our collaborators in the Richard James Lab at Seattle Children's Hospital. And they were very early adopters of nanovials. Nanovials allowed them to not only accelerate their research, but also answer questions that they weren't previously able to answer. So their lab is very interested in human plasma cells, and they had this observation that cells that are plasma cells by surface markers have this heterogeneity in the amount of antibodies that they secrete. And they had noticed this by ELISpot, but the ELISpot assay was an endpoint assay, and they weren't able to recover the cells to further delve into the mechanism behind this interesting observation. So with the nanovials, they were able to develop an assay that would allow them to delve further into this observation. Here you can see they modified the surface of the nanovial with an antibody to capture the secreted IgG from plasma cells they had differentiated from human patients. They then detected those antibodies with a traditional fluorescent antibody and used their existing flow cytometry instruments to analyze the cells and their secretion. And as you can see on the top, as the cells differentiate into plasma cells, they develop the surface markers. They have this heterogeneity again in the amount of IgG they're secreting. So this confirmed what they had seen previously by ELISpot. On the bottom, they wanted to, you know what they say, one picture is worth 1,000 words, or one imaging cytometry experiment is worth 1,000 analytical flow experiments. So they wanted to really see what this secretion looked like. And as you can see on this event, you have your surface markers localized to the cell, but the anti-IgG is localized to the surface, to the cavity of the nanovials, confirming that this really is secreted IgG. Once they were confident that they could discriminate between these different subsets of cells and quantitate this amount of protein secreted, they designed a new experiment very similar where they captured the secreted IgG and then came in with an oligo bar-coded antibody that is compatible with 10x Genomics single-cell sequencing. They then enriched their secreting cells by flow and then performed the single-cell sequencing assay. And here's just some representative data. Of course, you can read all about it in their publication, but they were able to correlate novel surface markers with these very high secretory plasma cells. CD59 was one of the examples, not previously described as a marker of plasma cells, but they found it very highly correlated by transcript with the protein secretion. They also were able to describe gene transcriptional networks that correlated best with this highly secretory phenotype. And this allows them to perform further experiments, probe further into the mechanism of this heterogeneity of their plasma cells. And this workflow is called secretion encoded single cell sequencing, or seq-seq. Here we have another demonstration of the seq-seq workflow. This time we'll move away from immune cells. This is coming out of Dino DiCarlo's lab at UCLA, looking at mesenchymal stromal cells, or MSCs. MSCs are super interesting. They're being studied for potential therapeutic applications in a lot of areas because they secrete a lot of molecules that are involved in various important processes, immunomodulation and regeneration. But the performance of MSCs in the clinic has been hindered by functional differences that come from cell source and heterogeneity of secretions, which points to the need for a method that allows you to describe the subpopulations of MSCs that are responsible for secreting the therapeutically relevant molecules. One of these therapeutically relevant molecules is VEGF-A, popular for its pro-angiogenic properties, and that's what they studied in this application. So they first used our EZM formulation of nanovials, which are compatible with capturing adherent cells as they were using MSCs. They also modified these nanovials with an antibody to capture VEGF-A and then used an oligobar-coated secondary detection antibody to be compatible with single-cell sequencing. And so we have some representative UMAPs of their data. And what you can hopefully appreciate right away is that, you have by secretion levels, these really high these clusters with really high secretion. However, when you look at the transcript, they don't always match. And so this suggests that the transcriptomic profiles are not necessarily a good predictor of what cells will be highly secreting of the protein. They were able to, using this sequencing data, identify a network of genes, which they called the vascular regenerative signal. And this was much more highly correlated with high protein secretion than the VEGF-A transcript itself. So allowing for the identification of these highly secretory subpopulations that have your therapeutic molecule, it unlocks the potential to isolate this subset for therapeutic applications. But it also unlocks the possibility of further studies on how you could modulate this secretion, also for therapeutic applications. Another pretty interesting thing that MSCs secrete are extracellular vesicles, or EVs. And similarly, the deployment of EVs for therapy has been a little bit hindered. And this is partly due to the difficulty in enriching cells that have high EV secretion, because again, you have some heterogeneity. In this workflow, which was also performed in Dino DiCarlo's lab at UCLA, the nanovials were modified to be able to capture and detect the EVs. So we use this anti-CD63 to capture the extracellular vesicles, and then a CD9 antibody to detect them. And this was found to be the best combination to be able to distinguish the EVs from the cells themselves. And so in this application, they used imaging cytometry again. And by flow, they were able to see this really high EV secreting population of cells and also this basically not secreting EV population of cells. And when they looked at the images from the imaging cytometer, you can see for the low secreting population, you really, when you look at CD9, you just see it on the cell itself. But when you look at this high population, you do see the CD9 signal all around the surface of the nanovial, indicating that it has captured these secreted EVs. They also, of course, sorted these different populations of cells out by traditional flow cytometry, again, using a Sony instrument. And these images are representative of what they saw post-traditional sort. So you still do see the differences in secretion in the high versus the low sorted populations. And just to take it one step further, they sorted out these populations of cells and injected them into a mouse model of myocardial infarction. And they saw that these were indeed functionally different. So with the highly secreting cells, you had improved cardiac repair in comparison to the control or the low EV injected animals. So with this demonstration, we were able to show that you can investigate not only secreted proteins, but also these secreted large macromolecules and isolate therapeutically relevant cells as indicated by their performance in the mouse disease model. So all of these applications I've highlighted are available to read more about in papers, and you can find these papers here and also on our website. You'll find our complete list of publications. And I encourage you, if you're interested, please check out the papers. There's, of course, a lot more detail and interesting experiments to be found there. We also have a lot of resources on our website. So we have our user guides, technical notes, application notes, where you can learn more about how the assay works, what you would need to do, what's involved. We also have video protocols for that. And we do offer virtual trainings for our established workflows. And then, of course, you're always welcome to email us directly, or again, check out our other resources on our website. And so with that, I believe we will open it up to questions and answers.
Thank you very much, Zuli and Katie, for this two very interesting presentations. So we're gonna go now, as mentioned, to the Q&A session. So feel free to type your question, and we will answer. So far we have one from Izzati. Is this approach compatible with cells from other organisms?
Sure, great. Thank you for that question. As demonstrated in the different application vignettes, we have performed assays with human and mouse. If you're interested in other organisms, rat or rabbit or something more exotic, we'd love to hear about it, hear about your interest, and see what we can develop and how we could work together to make those applications happen.
I'd also like to add that because the nanovials are highly modifiable and we also just provide options for purchasing nanovials by themselves, if you have antibodies against an organism that you have already tested or developed, you can also bring those in and use them with the nanovial system.
Thank you very much for your answers. Is there any other questions on the floor? Please feel free to ask. I would have eventually one. I would like to know, because I know when you stain, like the classical way of staining secretion is like by fixing and permeabilizing cells, and which kills the cell, etc. But it's a kind of stopping point in your protocol because it takes much longer to do it. I just want to confirm that there is no real stopping point in the workflow using the nanovials, right?
Sure, great question. So, you're correct in that, you're not fixing the cells, so you don't have that really stopping point after your flow staining. There are some stopping points earlier on in our workflow, so you can modify the nanovials up to a week in advance, and then you would just store them at 4 degrees until the day of your assay. So that is the sort of way to cut the assay into two parts. But because the cells are remaining viable the whole time, once you start your secretion incubation and secretion detection steps, you then need to move on to the sorting if you want to get the cells back at the end alive. If for some reason you were only interested in analytical flow, you didn't want to sort out live cells, you are able to fix the cells with traditional method, fix the cells in nanovials with paraformaldehyde, for example, and then analyze them by flow cytometry later.
Okay, thank you for your answer. So we have two questions here. So from Lin Wei Kang, do these nanovials needs to be emulsified? If not, is crosstalk an issue?
Okay, I can take this question. So I think this user may be referring to at some point we had a workflow step of emulsification. Right now, the nanovials don't need to be emulsified in the most recent workflows. A lot of this issue is solved by our EZM formulation, which has colocalization of all the biotin that serve for detection and cell capture within the cavity. So that really helps reduce some of the crosstalk. So this is not an issue for us right now. Of course, if you have something that's really high secreting, that's something that you may want to do some timelines to look at those different secretion times to see if this is an issue for you. But currently, we are not doing emulsification.
Thank you very much for your reply. And the second question from Han Jingwen. Would it be possible for us to identify the unknown cytokines directly, or must we perform first an ELISA to detect the different cytokines being secreted and then quantify them with the customized nanovials?
Sure. Thanks for that question. If I'm understanding correctly, it sounds like they might be asking, can you sort of do more of a discovery workflow, trying to figure out what cytokines your cells are secreting?
I guess so,
Um,
yeah.
yeah, versus so yeah, the assay for cytokines, and again, these are sort of beta kits that we give out on a case by case basis. But yeah, so far it has been we are looking at defined cytokines, so the interferon gamma, for example. Um, I'll have to get back to our technical team about, how feasible it would be to sort of profile more cytokines. Um, right now, the way the assay is designed, to be able to specifically capture cytokines, you would need to know what they are so that you can modify with an antibody and then also detect a fluorescent detection antibody.
Thank you very much for your answer. Would it be possible, like, let's say if you are interested in, let's say, 4 cytokines-ish, would it be possible to customize the nanovials to capture these 4 cytokines? Then by stimulating your cell, then you will know which one are secreted, for example?
Yes. So we have internally performed capturing 3 cytokines. It's possible to do more. Of course, there's hundreds of millions, but there is a finite number of binding sites on the nanovial. So as you keep increasing, you will eventually lose sensitivity.
Yeah, sure. Okay. So, thank you. Is there any other question? Yes. What would be the approximate number of biotin sites in the larger 50 micrometer beads?
That's a great question. I will have to get back to you on that after conferring with our technical team.
Okay. Is there any other question? I don't think we have any other questions, so I think we can wrap up. And so thank you very much for attending this webinar, and thank you very much for the Partillion Bioscience team for helping us organizing this event. And we wish you all a very good day and a good weekend.
Thank you, everyone.
Thank you.
Thank you.